Hey guys, let me tell you how I spent my weekend. It was a beautiful Sunday morning and I was taking a leisurely walk in the park. The trees were dancing to the wind, the birds were flying in the sky, and then there was my dog who was chasing after this rather beautiful butterfly. So I was sitting there wondering, what is it that is responsible for all the life around me? Then I realized it's just DNA, or deoxyribonucleic acid as we know it. All living beings, right from insects, plants, animals, and even as humans, contain DNA. Life would not exist without DNA. It pretty much dictates every aspect of our lives, right from how tall or short you might be, to the color of your eyes, to the color of your hair, down to the very interactions that take place between two molecules in your cells. This is kind of why DNA is known as the most detailed blueprint for life. Take the cell here. Cell is the basic unit of life, right? So at some point in its life, the parent cell divides to form two new daughter cells through the process of cell division. And the daughter cells would further divide again and again until they result in lots of cells that together form a whole living organism. But we already know that life cannot exist without DNA. So this cell should contain DNA, right? And when this cell divides, the daughter cells would also end up with the same DNA molecules. And by an extension of this logic, all the cells that arose from the original parent cell would contain the same DNA that was present in the parent cell. This property of the DNA molecule to be inherited across successive generations is the reason why we call DNA as the molecule of heredity. Or in simpler words, DNA is the genetic material that is passed on from parent to the offspring. And what we are going to discuss today is how it is that DNA came to be known as the genetic material. So DNA contains all the instructions an organism would ever need for growth, development, and functioning at pretty much every level of organization. You might have read in the news how scientists are constantly manipulating DNA in crazy ways to do wonderful things like making spider goats. Nah, this isn't possible yet. But scientists have succeeded though in getting normal looking goats to produce the super strong spider silk in their milk. They're even trying to grow human organs like liver, kidneys, and even hearts in pigs. Crazy, right? They have succeeded in even curing leukemia with the help of gene therapy. Such fascinating things that have been done with DNA. And there is so much potential for so much more. There was a time though when scientists didn't even know that DNA was the genetic material. This didn't mean that they were completely in the dark though. They had some pretty smart ideas, and thus Frederick Griffith came into the picture. Hospitals in the year around 1928 had many cases of pneumonia. It was a very serious disease, which would usually start with a cough and eventually lead to death. Since it was responsible for so much death during that time period, Frederick Griffith was working really hard to create a vaccine for that disease. So he already knew that the bacteria Streptococcus pneumoniae, or the pneumococcus as it was known, was the agent that caused the pneumonia. So there were two main strains of this bacteria, the smooth strain or the S strain and the rough strain or the R strain. The smooth strain was called smooth because of the smooth appearance due to the presence of a polysaccharide capsule. On the other hand, the R strain was called rough because of its rough surface, which was a result of the absence of a capsule layer around it. And of the two of these, only the S strain was virulent and was known to cause infections. And our brilliant scientists wanted to know why exactly were there two different strains of the pneumococcus but only one of them caused an infection. So he set up an experiment with some mice. In the very first scenario, he injected living R strain bacteria into some mice. And it turns out that the mice were alive. In the second scenario, he injected living S strain into a second set of mice, and the mice died. So far, these were the expected results. The R strain was non-virulent and hence the mice didn't die, while the S strain was virulent, which caused the mice to die. Moving on, in the third scenario, he took living S strain bacteria and killed them by heating to a very high temperature. He then injected the heat-killed S strain into mice and the mice lived. This meant that living bacteria were vital to establish an infection. And in the very final scenario, he mixed some heat-killed S strain with some living R strain and injected them into the mice. So what do you think happened to the mice? Well, the R strain is harmless, so there should be no chance of infection from that part of the mixture. On the other hand, the heat-killed S strain should also be harmless since dead bacteria cannot cause an infection. So we can probably make an educated guess that the mice in the fourth scenario wouldn't die. But surprise, surprise, the mice were dead. So what he did was he isolated the bacteria from the dead mice, and he found that there were only S strain bacteria in there. It turned out that even the descendants of the isolated bacterial cells were also the virulent S strain bacteria. He was flabbergasted. He thought about it deeply and eventually came up with an hypothesis that there was a factor in the S strain that survived the heat treatment and was transferred into the living R strain cells, which transformed them into the virulent S strain cells. He called this as a transforming factor. While he didn't know the identity nor the nature of the factor, he was sure that the factor was indeed responsible for the virulence, since successive generations of the bacteria were also virulent. This indicated that it wasn't just a temporary virulence that the bacteria had acquired, but was in fact hereditary. This hypothesis of a possible hereditary transforming factor was a revolutionary idea of that time. So many scientists got on the bandwagon of trying to find the identity of this transforming factor. It was nearly 16 years later, in 1944, that one such group of scientists of Oswald Avery, Colin MacLeod, and MacLeod-McCarty succeeded in it. They meticulously investigated the step in Griffith's experiment where heat-killed S strain bacteria were mixed with live R strain pneumococcus and then injected into mice. So this is what they basically did. They took a suspension of the S strain bacteria and heat-killed it. Then they separated the various fractions of biomolecules that were present in the suspension, which was basically nothing but polysaccharides, proteins, lipids, RNA, and finally DNA. Since they had to figure out which of these biomolecules was being taken up by the R strain cells, they incubated the R strain bacteria with each of these fractions separately. And whichever biomolecule would convert the R strain into the S strain would be our transforming factor. They kind of expected that the polysaccharide fraction would cause the R strain to turn into the S strain, since the presence of the polysaccharide capsule layer was what differentiated the S strain cells from the R strain cells. However, that wasn't really the case. In fact, none of the fractions resulted in S strain bacteria except for the DNA fraction. The R strain cells that had been incubated with the DNA fraction had resulted in some virulent S strain cells along with the R strain cells. And thus, the identity of the transforming factor was unveiled, and they came to the conclusion that it was DNA all along that was the agent that was responsible for whether the bacterial cell had a polysaccharide capsule or not, which consequently determined whether the cells were virulent or not. Now, in spite of the experiment being quite definitive in its results, scientists were a doubtful bunch. They didn't really accept the validity of this experiment. In fact, a lot of scientists continued to believe that it could still be the proteins which were the transforming factor. So how did scientists finally come to believe that DNA was the genetic material? What was the experiment that convinced them to let go of their doubts? Who were the scientists who answered the question for once and for all? The answers to all of these questions are going to be discussed in the next video. So thank you for tuning in.